Right, good morning. Welcome to the Pest Management virtual meeting. My name is Rebecca Stilton and I'll be co-hosting and facilitating the meeting this morning. And I'm the product manager for our insecticide portfolio. I'm also joined by Dr. Max Newbert, who's our expert in everything pest management, and he'll be taking you through the presentation this morning.
Good morning, everybody.
Thank you, Max. But before we begin, a couple of housekeeping things just to make you aware of. So you may have noticed we are recording this session, and it will be available on YouTube afterwards if there's anything that you wish to go back and review or share with any of your other colleagues. We thoroughly encourage questions. And you'll find a function at the bottom of your screen if you highlight with your mouse the Q&A function. If you want to send any questions to myself and Max in there, then we'd be more than happy to answer them at the end. And depending upon how many people we get on the webinar, then we may well go to raise your hand and you can ask your question yourself. But if there's too many, then it gets a little bit difficult to manage. There will be basis and Neroso points available, which will be sent out with it via an email, kind of within about 48 hours after the webinar, along with a little survey, which we'd really value your feedback. So without further ado, I will pass over to Max, who'll take us through the presentation. Thank you, Max.
Thank you very much, Rebecca. And good morning, everybody. So just as a quick agenda to what we'll be doing today, we're going to talk about sort of the lessons, learns and information we've gathered over the last season for barley yellow dwarf virus or BYDV, pressure that we're seeing, vector resistance as in the aphids, and then mitigation practices that we could probably approach going forward, as obviously we're going to another year without having neonicotinoid seed treatments on our cereal crops. Then we'll move on to cabbage and flea beetle, talk about what we've seen similar to BYDV in pressure and the resistance in the beetle themselves, and then what information we'll gather from our all-seed rape site this year. And then at the end, I'll just touch on winter stem weevil, pressures and timings and what we've seen over the last few seasons as well. As Rebecca said, do feel free to write into the Q&A as I go through, and then we'll address them at the end. But starting with BYDV and aphid control, just to give an idea of the level of control and pressure we've had in the last season. Now, when we come on to the aphid data that's been gathered at the Rothamsted suction traps, you'll see actually we had a relatively low level of aphid numbers caught, especially in the latter part of the season, as the season, like the previous one, became very wet and not conducive to aphid flights. However, that being said, at the start of the season, there was quite a lot of aphids, and I think it was a bit of a kind of autumn to start with, before the winter really set in. And what we saw, especially in the east where I'm based, this is Norfolk, this is some winter barley, so middle of September. And the area you're looking at on screen is the effect of BYDV when no application of hallmark has been made. This square here is from just a mist spray that's occurred. One application of hallmark happened to the rest of the field, and you can see the dramatic effect from having that first early spray has on reducing population expansion within the field of aphids. So a lot of fields that had, because of the season, timing and logistics, very difficult to get on, so either had no sprays, late sprays or limited sprays, all leading to loss of control this season, more than expected from the aphid numbers we were seeing. So you're not really going to be seeing this until the sort of new year if infection took place in the autumn. So it just shows that we really need to get the insecticides on in a timely manner, and also the amount, because the field also, as you can see around, there is areas of infection outside the mist sprayed area. So 2 applications would have been probably beneficial in this scenario. So to help give a sort of understanding of all the things you can do to get on top of BYDV, we've again still got our simple 5-point plan. So removing the green bridge, and especially if you're spring cropping and there's volunteers, make sure you've got a delay in drilling to optimize control before the new crop emerges. Is really key and crucial. We know that if you're glyphosating off an area, it'll take up to 2 weeks for the majority of aphids to vacate the green bridge and move on. So you want a nice gap between spraying off the green bridge and your crop coming through. The other thing is delayed drilling. Mid-October onwards, generally, depending on your area, will be in the sort of time period where then after that point, your risks will really dwindle down. We'll have a look at that in a moment. There is issues with that crop type, particularly with barley, and we'll touch on that with what we have generated data in the last few seasons. The other thing is optimizing establishment. So this is either seed rate or potentially seed treatment. I'll touch on vibrance, but primary infection when single plants are infected by aphids can be overcome or made the crop more resilient by having more plants per square meter, so that when a crop has a few plants infected, the rest of the crop can sort of compensate by tillering to that space, and that's aided by having a tighter amount of plants per meter squared. The other area is obviously we've got resistant varieties, both wheat and barley coming through. I'll touch on those quite heavily in this talk. I still think they will need an insecticide program, and they fit very well if you're drilling early in very difficult to control scenarios. You know, a field that's always had problems controlling BYDV, even when you had to turn, you know, you had to turn a couple of sprays of pyrethroids and you were still having loss of control. And I think that's where they fit well, because they add an extra layer of control to BYDV, but they are not necessarily the silver bullet. So they will still need managing, and also we want to make sure that these resistant varieties stay resistant and we don't have any viruses breaking that resistance. And then to aid, if you do need insecticides, the BYDV Assist app will still be here for this season coming forward. So that's an app where you can just put in your emergent state, your location, and it will keep track of when a population might expand of aphids in that field and warn you about that to allow you to plan your spray timings correctly. But just to give an idea for this autumn, you know, with different BYDV scenarios, variety and insecticide sprays with a few notes. I've sort of split this down. Let me just put my pointer on for yourselves. I've split this down to sort of 3 timings, and these are vague because obviously these will be dependent on where you are. If you're up in Scotland or down in the south, there'll be different dates, but early drill windows where you'll have obviously the maximum exposure to BYDV, the longest exposure to populations of aphids coming in and growing. This is the highest risk scenario. That's where I think, especially for barley and wheat, that's where the resistant varieties fit in. I mean, also you have to be thinking of the disease risk, you know, from our point of view, Graham and the like fit that, because you're increasing potentially rust and septoria. In the case of Graham, septoria being 6.8. So that's where they fit really well, but still, and you'll come to see why in a moment, they'll still need insecticide applications. And if you're drilling, say, early September, the likelihood is you'll need 2, maybe more insecticides across that crop. Just keep on top of the aphids developing through the season, especially if it's mild this coming autumn. Then if you're drilling that sort of optimum middle window, especially for our point of view with barley, that's exactly the perfect place for hybrid barley. And that's where we would say, with the yield you'll get and protecting it well with 2 insecticide applications, then moving on to a conventional variety or a hybrid variety over the resistant variety makes a lot of sense and returns the best, you know, returns on investment you have made with your variety choice. And then the later scenarios, you know, move into the conventional varieties, move away from the quality varieties, because what you're doing there is you're still hopefully getting the yield and you've got a higher seed rate, which helps with establishment when we're going into that November period onwards, especially with barley. And then moving on from thinking about what variety overall, focusing on the resistant varieties, we do need to understand what resistance means for barley dwarf virus. So with barley dwarf virus, we have different resistances in both barley and wheat. They are known as semi-resistant. So with our industry, it's probably more akin to how we understand tolerance. Because what's happening is it's not an extreme resistance, so infection will still take place, the virus will still replicate, but it will do it at a lesser extent. So if you tested the amount of virus in individual plants of a resistant versus a non-resistant plant, you would have more virus in the non-resistant plant. And the knock-on effect of this is obviously the plant is less effective because there's less virus being a parasite on the plant and taking resource away. But there is nuance. So I've tried to make this as simple as possible, but it is a complicated matter where you've got different viruses involved, different vectors with different prevalences, and then the effect of the genes might be slightly different. So there's 2 main types that are thought to be in the UK of barley dwarf virus. MAV, which is predominantly spread by grain aphid, we'll come on to is resistant. The grain aphid itself has some resistance, heterozygous KDR, and that's thought to be a sort of milder form of barley dwarf virus. And this type of virus is endemic in the country, so it's everywhere, but you will have reduced titer in both the wheat and barley resistant strains of this. So that's very good news. With PAV, this is thought to be the slightly more prevalent, potent BYDV strain. This one's transmitted primarily by Bird Cherry-Oat Aphid. As we'll come on to later, this is a bit more numerous or less trap shy than the grain aphid. This has got yellowing and stunting symptoms, again common, and will get good effectiveness of both in barley and wheat. There's a RPV strain, which is actually a slightly different virus. It's not a luteovirus, it's a pleurovirus, a sort of close cousin to barley yellow dwarf virus. And this is a cereal yellow dwarf virus. Again, we think it's predominantly spread by cherry-oat aphid. It's common as well, and we'll get onto how common in a second. But the thing to know, the titer does get reduced in the barley form, but not to the same extent as the other two, because it's a slightly different virus. So this is something to watch out for, but for the wheat, we still have good levels of reduction in titer. The added complication, and this is just for East Anglia, Cambridgeshire, Norfolk, Suffolk area, what we have is a wheat dwarf virus. Now, this is a completely different virus because it's a DNA rather than an RNA virus, totally different structure. Also, it's spread by leaf hoppers, and this will primarily exacerbate or express itself as primary infection, and might look like secondary infection near the headlands of barley yellow dwarf virus, but because this is a completely different virus, there is no resistance to this type in the barley or wheat. So this is potentially a watch out going forward. So that's more of a Eastern thing, but it's to be aware of that. So if you're seeing symptoms, it might not be barley yellow dwarf virus, it might be another form of virus in your crops. But with this in mind, we did some work, and we're continuing this work this year, but obviously haven't got the yield data, but we're doing different varieties and different drill timings. So looking at November and October drill timings. And the first thing to say is, obviously we were planning potentially more September, October, but with the seasons we've had, it delayed everything. And when drilling late, especially with barley in this instance, it increases the lodging risk as well. So drilling in optimum timing also helps lodging, let alone BYDV and the impacts on that, and grass weeds as well. But when we're just focusing on King's Barn, which is obviously our 6-row hybrid, and one that we see as a very good standard to compare things against, we tried the Amistar variety, which is a resistant variety for the YD23 gene. And we did a October drilling, November drilling. In the October drilling, we had non-infected that had a full insecticide program, and then we did an inoculated trial with virulifera's aphids just to see what the impact on barley yellow dwarf virus is. And you can see that the impact of barley yellow dwarf virus happened to both varieties. Now, I think Amistar is the first generation resistant variety of barley, and I think it doesn't quite have the vigor of other varieties in the RL. So as time goes on, I think this will be very much like the all-seed rape, where the first couple of varieties will be interesting, but as time goes on, they'll get better and better, and the background variety will be much more suited, because barley yellow dwarf virus causes a stress exacerbated symptom, really. So if the variety is not very vigorous and doesn't cope with stress well, especially in these drill timings that we had in 2020 harvest, which is 2019 autumn, what we're getting is a very stressful season, virus infection still occurring, and we're still getting these impacts even on the resistant variety. Now, as time goes forward, I think the resistant varieties will have less impact because the variety itself will get better, and the resistant gene is really important and will be really helpful. And then obviously you can see the impact in November drilling hit both varieties. The thing is, what we want to see about this, what was best, to drill in optimum timings or avoid drilling? What's the biggest penalty? So if we look at sort of what this showed, when you compare King's Barn to Amistar at full potential, so this is no BYDV, in this trial, obviously this is one trial, we were 1.1 tons ahead. If we had infection take place, we were 1.5 tons ahead. So it's about picking the optimum variety. I think this was drilled in early conditions. In nicer conditions, the Amistar would have done well, but I think it really should be used in that very early period in fields where you're having difficulty controlling BYDV, and historically. Because if we look at what the impact of BYDV will impact November drilling is, you can see with the BYDV impact, we had about an 8% yield loss in the King's Barn. In the Amistar, we had 14% yield loss. But if you look at the November drilling, for both, the November drilling in most cases had a larger impact than the BYDV, maybe not so much for the resistant variety there, as you can see. So the key thing is choosing the best timing and protecting that variety. And I think this goes to show, and it's the same for Wolverine, infection can still take place, yield losses can still occur. So we want to protect that variety. It's not to replace insecticides, it's just to help. So with that, I would have a full program still over a variety, especially if you're drilling it early. And just to put that in numbers, what the average yield loss for the two types of mitigation factors are, BYDV there, and then November drilling. So with that in mind, and just thinking of how you have to control BYDV and how long for, this just shows over the season when aphid flights are occurring. This is a sort of combination of Bird Cherry-Oat, grain aphid and rose grain aphid. What we're having is potentially from September, the peak happening at the end of September into October and then going down with maybe 3 to 4 secondary spread events. So that's when the aphids have enough time and energy to build their population up and expand out from certain points. So with that in mind, when we look at those aphids we've been catching over those certain years, we've had 2 years of this now with Syngenta UK yellow water traps. We've got BYDV incidence over the autumn period. So to give an idea of what level of BYDV we've got in aphids, starting from sort of late September into October, we've got 2019 in orange and 2020 in blue. Now, the reason there's a bit of break in the blue 2020 line, we didn't have as quite many data points, but I put them on the same graph for comparison. But you can see the old rule of thumb used to be about 5% of aphids had BYDV infection. And on average, when you look across the season, we have about a third of aphids actually carrying BYDV. But we can go further than that, and we actually looked at what type of BYDV there was in the aphids in those 2 seasons. So what we had was 2019 actually was made up of that cereal dwarf virus. If you remember, I was saying slightly different to BYDV, that made up 72% of the aphids that carried BYDV. So it was actually the majority of it in 2019, and then MAB-PAB made up 28%. 2020 sort of flipped around, and we had 31% of RPV in 2020, and then that went to 70% of MAB and PAB. So it does obviously change over time, but all these are major viruses now in the UK, and we have a high level of infectivity of the aphids themselves. But what is interesting, if you actually look at the aphid numbers, I'm just going to focus on Bird Cherry-Oat aphid because they're the most numerous. You can see the red line is the Bird Cherry-Oat aphid numbers. If you look at the scale here, very large numbers potentially being caught in the autumn, over the autumn period. And you can see it was actually one of the lowest years we've had in catches. You can see in sort of bright green, that was the previous year, 2019, and that was much higher, even though we had a lower BYDV infection rate. And I think that was just how awful the season was. That amount of rain just kept them out of the crops. But if you look here, sort of early season, we had sort of a peak that was higher than 2019, probably had aphids coming in mid to late September, populating crops, and because we didn't have quite as horrendous rain, they did manage to propagate in the crops and spread BYDV. So that was the kind of pressure we've seen. So obviously it's to be determined yet what the kind of pressure will be going forward. But if we have early invasions of aphids, it seems like that will be adding the pressure. We know year on year we've got about a third of aphids carrying BYDV. So next is sort of keeping track of that. So for ourselves, we've got the website syngenta.co.uk/BYDV assist. Now that page has information about our water traps, and we've got 13 sites for the UK, so you can keep track of that live. We'll update that weekly, but also it links through to our BYDV assist app. Now this is a free to download app, and all it needs, as I said previously, is basically your location, your emergence date, and it will keep track of each field you put in, and it will say when there's enough day degrees accumulated for a population to expand. So that will expand, and if you don't control it within its risk window, it could potentially have those large area effects of infection. So I would strongly suggest downloading it. It also comes with a spray guide, so if a warning comes up on your phone, you can then look ahead the week coming to see when there might be a good spray window to adjust when you'll be spraying. And that's just secondary spread, that's not for primary infection. But then that sort of moves us on to spraying for aphids, and what do we know about resistance? So we know there's some evidence there might be a slight tolerance now happening to pyrethroids in Bird Cherry-Oat and rose grain aphid. Nothing significant yet, but it's not a surprise since they're rapid cycling aphids on a very numerous or large hectareage crop being exposed to pyrethroids. But at the moment, no real change in Bird Cherry-Oat or rose grain aphid. However, in avenae, Cytobium avenae or grain aphid, we know there is heterozygous resistance, and this is a KDR point mutation resistance, which makes in lab tests them 50% susceptible, but as we'll come on to, in field at time of application, still susceptible to pyrethroids. We did have this from Rothamsted, Martin Williamson and Steve Foster been testing their aphids, and you can see 2020 average, we had about 20% of the aphids with resistance. 2019 was a slightly lower, about 12, 13, 14% potentially. So it has come down a lot, because last time we did this, which I think was 2014, we had as high as 70% or 100% of populations infected on, average or more often than not. Now you can see the only one that really stands out there is York in 2020, where we had a very high level. So that's a bit of an anomaly, but generally the resistance has come really far down, and in many instances is less than 5%. And I've just tried to put the 2 years, 2019 and 2020, separately, and then I put the sites that we had tested over the years together. So you can see there isn't necessarily a pattern to resistance between the years, and it seems to be low level. So there's some fluctuation to the resistance. It seems to be going down generally compared to what we've had previously. So that fitness cost that is associated with resistance is having an effect on the grain aphid. It's not become like resistance in mysis, which has become the majority of the population. But then just moving on to talk about resistance and the work done by Alan Jur. So you can see the citation at the bottom there, did some work where he looked at resistant and susceptible grain aphid. The dark bar, blue, dark orange bar being this fully susceptible to susceptible alleles, or on the right hand side bar, if it had the KDR heterozygous resistance. Now we know from Steve Foster's work, we get about 50% control of resistant populations. However, the 50% that survive in the lab tests are in ideal conditions and not obviously controlled, but they will be affected because they still have susceptible sodium channels. So in the wild, they're not fit and they will perish regardless. So when you see this, then you get pull out nuances between the different pyrethroids, and we still have lambda cyhalothrin and hallmark giving that really high level of control that we've seen, compared to cyper and deltamethrin, a significant boost over them. So we know it's a more potent pyrethroid and does give better control. Now, obviously, people will say all lambdas are the same, but year on year, we've done work with Alan Jur as well, looking at generics versus hallmark. And we know that the formulation of hallmark is encapsulation, its suspension, and then its ability to degrade slower, because the sort of implications we have with the titanium oxide in there, we know it has a greater persistency. And this does lead to increases in control. And generally, we see 20 to 30% increases in control when using hallmark, just purely because of the formulation, let alone the same level of AI in the product. The only nuances here was in this year, we did activator 90, and this had actually quite a high level activator 90, and I think we did 1% rather than 0.1%. And this, although did increase the level of control of aphids, if you can see that this was the control of aphids, this graph, when we looked at the virus the next year, the hallmark versus the hallmark with activator 90 that had the high percent, it actually gave higher levels of virus control. And this might be due to the fact it's acting a bit like the oils and potatoes, where it's blocking up the stylus. So we plan to do more work on this going forward, because this will be really useful. So sort of to sum all that up, BYDV is a risk, especially if we get that early aphid flight. It doesn't have to be huge, but if they get into the crops, into those early drill scenarios, that could be a problem. I think fields that are surrounded by a woodland in a lay of a hill, coastal regions that year on year get virus problems, that could be a really good fit for the new resistant varieties coming through, both wheat and barley. But as we've seen, we will need to protect both those still with an insecticide program. So you can see on the screen in orange where we put hallmark. If you think back to one of the original graphs I put together, or tables, sorry, the earlier you go, obviously the larger the insecticide program becomes. So it is to be aware of that, using the BYDV Assist app to put your fields in to keep track of it and make sure that everything's got timely applications. It is a conservative model. We're assuming that aphids land in the crop as soon as it emerges, but that means you will get the best effect, especially in high pressure areas. And it's not until late November, if you really potentially might not need applications, but again, that does have some seasonality to it as well. And just before I finish on BYDV, obviously I've talked about BYDV, the vectors, resistance, control, but the other thing is that, increasing establishment. And that's where VIBRANCE Duo, although this is a seed treatment we've had registered for a few years, has nothing to do with insects, or it's not an insecticide, it's Sedaxane and Fludioxonil, 2 fungicide treatments. It will have a benefit on vigor. And what we've seen, and you can see on screen, is no matter the scenario, plough, direct drill, especially those difficult scenarios where you may be late drilling, it does increase plant establishment and tillering. So thinking about pests and, outside BYDV as well, fly pests, if you increase tillering and plant numbers, this gives you a more resilient crop. It can accommodate more losses due to the fact that there's more plants down there. It's a good extra layer to protecting the crop and making sure you yield at the end of the year. The other thing to say is it's got full approval now in barley, so you can use it in malting barley. And we know, again, it has the same increases. So this is some King's Balm, but we were seeing, up to 23% over establishment over and untreated, 13 over at Vauxhall Star, and then that increase in tillering again. So I think this is a good addition, although nothing to do with insecticides. It's just a good practice to make a more resilient crop in difficult scenarios. So that's everything around BYDV, quite a lot to go through, but I'll finish up on cabbage stem flea beetle. Now, again, we've got a 5-point plan, and I think hopefully we'll go through most of this to show it's quite key, but really year on year, we see light till assumptions best. Generally, we want to conserve moisture in the seedbed. I think that's one of the biggest factors for establishment rather than cabbage stem flea beetle. That's a large one. But if you don't have moisture, and you don't have moisture coming, and you have a lot of cultivations, you release the moisture and then try and establish a crop in a dry seedbed, we're not going to have even and good establishment. Next to that is consistent depth, vigorous seed as well. I'll come on to the 1,000 seed weight in a second, but consistent depth will make germination happen very evenly, which will reduce the chances of uneven germination and cabbage stem flea beetle coming along and taking off a few plants at a time. Talked about moisture. Now the main thing is thinking about 1,000 seed weight. That gives extra vigor. We've had at the site a conventional variety and a hybrid variety, and the conventional variety was always behind the hybrid through the season. It was better to establish a larger plant and go through the growth stages a bit quicker than the conventional, the hybrids. That extra vigor early in the season obviously is a benefit to when you're trying to establish and grow a crop of all seed rate. And then finally, if you do have issues with cabbage stem flea beetle, it's still using the best pyrethroid product on the market, which is Hallmark, if thresholds are met. So that would be our 5-point plan, and we'll go through the nuance now. But just to talk about pressure, I've put together an cumulative graph. So this graph shows all the data from our water traps from, well, 2016, 2019, and then in green, that's 2020, the season just gone. So this is the autumn period, going from sort of end of July up until November timing. And you can see the scale there up to 3,000. And the year we were getting close to that was in 2019, which was one of the horrendous years for cabbage stem flea beetle. Early stage, one, because the numbers were so high, but also we had a very dry period during that year, during all-seed rape establishment. Obviously, it all changed later, but that caused us to lose actually our all-seed rape site that year. The year just gone, the autumn was actually very good, and that comes down to larval numbers. Because the autumn was so bad, the larval load was actually much reduced in the all-seed rape. So the subsequent year we had a lower population, and you can just see that was one of our lowest years on record. And we at our site got moderate pressure, but we didn't have much plant loss due to cabbage stem flea beetle. Then talk about resistance. This is from Rothamsted, but they've been doing sampling for many years, but this is 2018 to 20, which is the sort of most up-to-date samples. You can see the resistance status color-coded to the location on the screen, but really maybe the key bit is obviously 2019 was sampled very heavily, and the other years not so much. But generally, as anything with resistance and selection pressure, we are going up in resistance. So the year we're going to talk about the trials we did this year is in this year where we're getting close to 70% of the flea beetle population having some form of resistance. So as we know, there is resistance in the population due to the selection pressure we're putting under. So with that in mind, I'm going to talk about the results we've had from our 2020 drilled site, which obviously is going to be harvested shortly. But we had our site at Letton in Norfolk this year, so not too far from me, and that's where we're going to talk about the results. Now, the site, we tried to do a few different things. So on the screen is a picture of one of our small trials, but we had field scale trials with Chris Eglinton at the site, as well as our small plot trials. And this is an alternative establishment trial where we're trying straw, obviously sort of farm manure approach, and then we also drilled a companion crop, which you can see in this screen. And we were using berseem clover and buckwheat as the companion crop to try and mask and help the crop establish. The one caveat is all that was done the day after drilling, because obviously we were going into Chris's field after he'd already drilled the rake. When we did the field scale work of the companion crop, we did a lattice across fields. We were doing it a week beforehand and broadcasting it over, so it had time to establish before the rake got up. And just a visual of the site, we've got from left to right going through the season from sort of mid-September all the way into November. But the previous year, why we were using straw was because we had really good establishment with straw, keeping the moisture in the crop. But this year, we had more or less constant rain as soon as the site was drilled, so it acted as basically a blocking layer in the straw. And you can see that being a thinner part of the crop all the way into November. It did recover eventually, but it's not maybe a standard approach I would approve. We were also using wheat straw, which is thought to have a better effect on reducing flea beetle interactions. But that might have come through more in the larval studies we looked at. But that just gives you a visual of how the site develops. But as you can see, quite good establishment overall. Then to give you an idea of application timings before drill timings, application timings before we get on to the results, we had 2 fields. Any field scale work I talk about generally is from the 29th of July drilling, and we had the companion crop broadcast a week before that drill date. Then the small plot trial work, which was shown in the previous images, was drilled on the 12th of August. And then we were doing the things the day after, establishing the companion crop, putting on the straw, putting on the manure. We were using pig farm manure. Then you can see what we've got on the scale is the number of flea beetle on site over this period. And you can see we had very low pressure at the start. As we had germination, we had our first sort of increase in flea beetle to any large number. That's when our first application went on, basically just as it was coming through the ground. And then we had a second around the free leaf stage, which was just before the population exploded on site for the pressure we had this year. Now the results I'll go through, but there's 2 halves, so if you can draw a line down the centre here. To the left, we were looking at trying to get the most out of hallmark. Can you add things into the tank to try and increase the efficacy of hallmark? We either had 1 spray as a standard or 2 sprays. 1 spray gave about 50% control. 2 sprays gave 62% control. That first spray gave the most control, the best benefit to the crop. The second spray did help, but obviously protecting a crop when it's younger and more susceptible was more important. So everything is done off this 2 spray standard, and then we're adding adjuvants. So activator 90 is generally our advice. I think we're using this in a small plot trial with trialists going at the perfect speed, at the perfect height, in perfect conditions, and it didn't make any difference statistically. So activator 90 in a boom sprayer through a tractor might have a better effect, whereas things like oils definitely had a deficit. So wet sit and phase 2 are oils, and I think what's happening there is you're causing penetration of the AI into the leaf, because it's allowing penetration of the lambda into the leaf, taking it away from the surface and coming into interaction with the pest. So in effect diluting the effect of the insecticide you're putting on. So anything that aids penetration is not to be used with contact insecticides. We also looked at pod stick, thinking there's been a lot of talk about potentially that interacting physically with the cabbage and flea beetle, either gumming them up and making them unable to move or chew, or creating a protective layer on the crop. As I thought, this is basically PVA glue. You're locking up the AI in, you're reducing the amount of interaction the insect might be having with the product, and so you're having the largest deficit to interaction with the product. So that would definitely not be advised. BLA is a latex type sticker, but is used with herbicide and doesn't lock up the product, and that had the same effect as activator 90. So not necessarily recommend that, but as long as the product doesn't lock up the AI, spreads it as evenly as possible, because obviously contact insecticides, the key thing is getting good coverage. I think anything like that will do. Otherwise, using just the product with very good application techniques is the way to go. Then the second part of this trial is what level of control we get over the untreated with different things. So with the companion crop, obviously drilling a day later, the crop still came up. This is without an insecticide. But obviously there's a bit more competition there, potentially small plants at the start, and actually we had a deficit to control, you can see we were under 20% worse than the untreated because we had potential competition at the start. And I think that's something to be aware of, especially with bullish things. If you're thinking about using mustards, it does need to be aware of. And then with the companion crop and hallmarks included, so you had both the insecticide and the companion crop, that was actually the standout treatment on site, and you can see it quite clearly, where we got about 72% control. Now, the reason for that was because the first insecticide went on, it gave time the crop to not be attacked by the beetle. We had better control early on, allowing then the cover crop to grow up and protect the crop itself, and then we had the benefit from the companion crop without that early damage. Then with the pig muck, we had again a deficit to control due to it probably causing smaller crops, smaller plants, because it was acting as a blocker, and it wasn't early enough on, obviously, to act as a benefit to nutrients. We were hoping the smell would mask, but the key takeaway, I wouldn't recommend maybe using digestate or anything like this, is because it's difficult to time. You basically have to put it on exactly when the migration is happening, because it breaks down so rapidly once spread that the smell gives such a short time of control to mask the crop. And then what we're seeing also with organic amendments, both straw and muck, is it has a negative effect on the products you put on afterwards, because you're putting an organic layer over the top, which has two effects. It might shield the beetle from the insecticide, and it also might break it down very rapidly, being organic matter it's coming into contact with, rather than obviously leaf material. So that just returned it basically to the untreated. With straw, you can see it interacts in a similar way, where we didn't get the benefit of the straw because it was acting as a barrier, smaller plants, increasing the damage, and also the hallmark was getting directly intercepted by it, not allowing it to really interact with what it wanted, either the target or the crop. And just a visual there, you can see the untreated, where we've had two hallmarks. You can see the hallmark plus the cover crops being really lush and the standout. The pig muck, at this stage, I think the pig muck was actually adding a bit of nitrogen, and we've got slightly larger plants at this point with the pig muck. And then the straw, you can still see had an effect. So I think this is a sort of late October photo, or early October, late September photo. So it just gives an idea of what we were seeing on site. But overall, anything organic put on does need to be thought about with how it interacts with then your chemical control measures over the top. We also did the companion crop, and this is without insecticide. So this is field scale, where we broadcast a section over the week before, and we did it on the 29th of July drilling, and then we also did it in the same field as the August the 12th drilling. And as you can see, this is damage control. This is adult damage control without insecticides, and we had quite a large effect on reducing the damage. And this was because we had that crop established, a companion crop established a week before the rate. It got up, caused that interaction where it masked the volatiles of the oilseed rape, and we had this reduced attractiveness. So there was still damage, but we were getting good levels of control. So if thinking about a companion crop, make sure you get it in early so you get the maximum benefit. Then going back to the sort of companion crop trial with obviously the muck and straw, we went back and did a larvae assessment where we were taking 10 plants per plot, and we were then doing a sort of funnel experiment where we were then waiting for the plants to desiccate and then counting the flea beetle that exited the plants. And what was quite interesting was so the bars are the number of larvae we had for 10 plants. So roughly on site, we had about 13 to 14 larvae per plant. The numbers above the bars are the percent control. But if you look right across the board, anywhere where hallmark was applied, we had between 30 and 40% control of larvae. And this was at adult timing. So this shows two things. One, that the adults are ready to lay as soon as they come into the crop. They feed on the way. Literature thinks, we get them coming into the crop, then they feed for 2 weeks, and then they'll lay. Then we have 240 degrees over 3.2, and then they'll hatch. But I think we've got it all happening much earlier. We've had 2 insecticides really early on. That's protected them from the adults and the egg lay, and that's had an effect. The other thing is obviously we've got the companion crop without insecticide, reducing it by 16% the amount of larvae, so that had an effect in this trial. And then the straw also had an effect. That wheat straw did have a confounding effect on egg lay. The muck had no effect. It was very similar to the untreated. The other thing to note is that in the muck and straw plots, where we didn't have hardly any control, or even in some cases negative control of the adults, either it's having enough effect to kill off some to reduce egg lay, or what's more likely is that although it's not maybe at the levels to control the flea beetle, they might be resistant, as we saw in the previous slides. The repellency effect from hallmark is making them less likely to lay eggs. And even where we didn't see good control of the adult damage, we're still getting good levels of control with the larvae. So we had a 40% reduction. So in years where you have that cold start, frost, and potentially a sort of concentration of larvae in the plant, this could be really beneficial. And obviously, as I just say again, this isn't that usual October spray timing for Cabbage and Flea beetle larvae. This was just from the adult control timings. So that was really interesting to see. Then we looked at the larvae in the companion crops. We had a 10 kilo rate, a 25 kilo rate. The more companion crop you had on the crop, the better its effect. But again, you can see we had relatively similar, no insecticides in these treatments, but this was a field scale work. You can see we had over 20% control and up to just about 30% control, depending on the percent covered crop. 10 kilos more the recommended rate for this mix of Bertie and clover. The other thing we did on site was sheep graze, but we did it for 1 week, early September, and that did provide up to a 14% control. Now, I might not necessarily recommend this as an approach. It's a small gain for a lot of potential work. And also in previous work, if we have a poor spring following sheep grazing, the crop can't quite recover, and usually we lose more yield to the grazing than we do to the larvae. So that one's a bit tenuous to recommend across the board, but the companion crop is quite easy to recommend. The other thing to say with this mix is that, obviously here's a picture of it in the small plot trials. This was it in sort of the February timing, which gave us very nice control purely from frost and the winter. So it doesn't need extra chemical control. The other thing, it does leave these tufts of buckwheat stacking up like canes, which I know from experience will put off pigeons landing in crops. So if you've got poor establishment or a gappy crop, it could help reduce pigeon damage as well, just from the sort of desiccated stems of the buckwheat. So we had that both in our small plot trials and companion crop trials. From our point of view, it's a simple solution and quite cost effective. So moving on from that site, we also have a site at Loddington and Lenham where we're doing conservation agriculture project, where we're looking at low tillage or sustainable practices in a normal UK system. That generally boils down to a light till, min till and plough being 25 centimetre inversion tillage, min till being 10 to 15 centimetre non-inversion tillage, and light till being in the sort of top 5 centimetres at most. So sometimes direct drill, sometimes not. It just depends on the season and the use case. But generally that's the three systems, and we're looking at this across the board. But what's interesting for oilseed rape is we can look at those three systems and what's its effect on oilseed rape establishment. So this is Lenham this year. This Nathan Morris from NIA took these photos. You can see the plough plot has very poor establishment. Just to say, this is in sort of the March timing. That is the min till system. So you can see we've got better establishment. There is some plant and crop there. And then you can see the direct drill system. So again, this comes back to what I was initially saying about conserving moisture, especially on light land, doing direct drill, trying to get as much conservation of moisture. Also, rolling does help with oilseed rape very greatly. So if you're seeing the first 24 meters of the headland establishing well and not the rest of the field, that gives an indication as well. But when we look at the yields, this is the 2 years over 2 sites average tonnage between those systems, and we're basically getting a 15% yield gain from just light tilling. I think after this year, which was the photos you just saw, that's going to be a bit more dramatic, that difference, because obviously we've had poor to no establishment in some of the plough plots. But it's just to get a point home about as light tillage as possible so that we have better establishment. So just to sum up the Cabbage and Flea beetle, we've obviously got our thresholds. From our point of view, we were getting better results when you that initial control at cotyledon stage, protecting them when they're young and susceptible to damage. And obviously we've got to stick with the 2 rates. We were using 75 mils a hectare for the flea beetle, which we did have on the site. They do tend to come in slightly earlier as well than the Cabbage and Flea beetle. And then you can see our application advice, sort of 100 to 150 litres, and 3D nozzles alternative backwards and forwards. So that's our advice around application. With the BYDV much more towards that 50 mils per hectare. That would be our top rate for cereals. But with that said, I'm just going to touch on a few last bits before we finish up. I realise we're coming to the end of the talking piece, but I thought for especially the northern growers, but also in the south of the UK and the Midlands, we're seeing a lot more rape winter stem weevil this year. So predominantly this will invade at the sort of end of September into October. It will need sort of 4 weeks in the crop of feeding before they'll lay eggs. So you're predominantly targeting the adults before they'll lay eggs, hopefully. They'll lay, unlike flea beetle, they'll lay the eggs on the petioles and then they'll burrow into the plant straight away. So that's why we're really trying to control the adults. You can tell the larvae different to the Cabbage flea beetle larvae because they're usually a larger grub, slightly browner head. Easiest way, if you get a lens, if they've got legs, they're a beetle, so they'll be Cabbage flea beetle. If they've got no legs, they'll be a weevil, so they'll be a Cabbage weevil. And usually they've got a bit more serious symptoms where they'll stunt the plant very early on, compared to the flea beetle will just weaken it over time. But this is it from 2019. We've got some data from our Warwickshire sites, and you can see the dates at the bottom, but you can see the end of September 27th onwards into early October. That was the first peak, and then we had a few peaks into the October timing. So spray timings for us will fit sort of into that usual Cabbage stem flea beetle larval timing. Will also control the stem weevil. That was 2019. Moving on to 2020, we had those two sites, the early drilled and later drilled sites. So the early drill site was field field 1 was the later drill site, and field 2 was the early drill site. And you can see that we had different times of invasion for the two fields, but again, very much mirroring what we had in Warwickshire, where we had that sort of very end of September, first lots of weevil came in, then we had a small bit of activity, and then this was much later, but we were going into sort of early November where we had the second lot of larvae, adult weevil coming in. So slightly later, but again, if you're on this sort of control period, especially when you get a week to 2 weeks good efficacy with hallmark, you'll get good control in that October period of weevil. And just to show some data from 2016, cypermethrin versus hallmark, just note obviously the efficacy 55%, but we do get relative control with lambda. I think it's much more about the timing, but we know lambda is still a potent product against the weevil adults. It's just about good application. You see the application timing there was around the 26th of October, so we might want to go a little bit earlier before some egg lay happened. So just to summarize, before I do a few finishing bits on hallmark, from what we found this year is that we know that BYDV is endemic. It's across the country. We have high percents in the aphids. A third of aphids will have BYDV of some form. We know the RPV strain of cereal yellow dwarf virus is more common than first thought. Some resistant varieties are not as fully resistant to that as other forms. And all forms of the resistant varieties we have for wheat and barley now are only semi-resistant, so we need to protect that as long as possible. So we still need a full IPM solution, so that's still an insecticide over the top, especially because they'll be put in the highest risk scenarios. So the best yields from us are picking the right variety for the time. So very early, difficult scenarios, pick the resistant varieties, optimum drill timings, choose especially what fits your market best end use, or our highest yielding. So for barley, things like SY Kingsbarn, or in wheat, obviously we've got now an extremely high yielding variety, SY Inciter, which also has a 6.8 for Septoria and orange wheat blossomage. That fits very well, especially on light soils where we might have more issues with aphids. You choose those in the optimum times to produce the most yield, but protect them through a good insecticide program, help planned by our BYDV Assist app, which is free both on iOS and Android. Just have to go on those relative app stores and search it and download it for free. Then for Cabbage and Flea Beetle, again, the message is low tillage, conserving moisture, good establishment, even establishment is what you need. So consolidation, even drill depth, that's what really helps at the start. Then if we are thinking about the larvae, hallmark still is the best product we can use. Yes, resistance is an issue. As you see, the results we had this year are from that high level of resistant population. We know that we're getting good control, and it's really, if you want control of hallmark, it's using it as well as you can by not putting too many things in the tank, good application techniques, and that will give you the best chance of control. And you hopefully will also have that knock-on effect we saw this year, which was the residual effect, having a reduction in the larvae as well as the adults at those early timings. But just to get the importance of hallmark, and this will matter still in the autumn, is that what we have in the formulation really does help. This is a high UV continuous lamp test where we put products under a lamp, UV lamp, which will simulate the sunlight and degrade products. And you can see the orange line being hallmark and the blue line being a generic lambda with no titanium oxide in the mix. It doesn't have that sunscreen effect, and this here says 50% remains more active after sort of 3 days of continuous UV treatment. But probably what's more important, because the generic lambda is fully gone by day 3, is to look at day 1. And actually we have sort of a, from 20% left for the generic, we still have 80% left with the hallmark. So, you know, you've got a factor 3, 4 there of protection. So that really will extend the control period. And when we talk about these pests, which will have long windows, even in the autumn where there is, you know, there's still good sunlight, especially early on in August, if you're doing early timings, that'll be really key to get control. And then just to show the physical difference that there is with the formulation, especially around sunlight, you can see what happens. Hallmark will not split out in direct sunlight because of formulation, whereas with generics we do get a split. The split also occurs if there's long-term storage. You can see there is different formulations here of different generics versus hallmark. Hallmark, after 1 year, no mixing in storage, will still look consistent because we've got a good suspension formulation holding it evenly in the bottom no matter how long it's there, whereas you might get some bottoming out in other formulations. So there is that physical and chemical difference which helps that control we see with hallmark. So that is really the sort of quick fire summary of the sort of major pests we're thinking about this autumn and the learns we've had with hallmark and how to get the best control. It's basically just to be aware, do timely applications and just make sure you're keeping what the season's doing, and that will allow you to get the best control possible. So with that, I'll finish up sharing there and pass back to Rebecca if we've got any questions come in.
That's great. Thank you very much, Max. We've had a question come through on the chat. If anyone else wants to ask a question, then please either put it in the Q&A or raise your hand and I can unmute you to ask it directly. But the question we have is from how long do you anticipate hallmark will keep giving 30 to 40% control of cabbage stem flea beetle?
So I assume if you're saying 30 to 40, that was talking about the larvae. So with the larvae control, obviously that would be hard to say. That was mostly seems to be from residual effect. It also reduced it down because the amount of adults aren't there to lay eggs. But what it seems to be from the results this year, it was more from the fact that we were getting the repellency effect, where the adults might still be able to feed and not be controlled, but they're getting irritated by the pyrethroid enough to not to want to lay. So difficult to say. Obviously that level of control could potentially go up if we did a larval timing application, but this was done in a year where we had sorry, that's my cat just come up. We had a year we were doing in 2020 autumn where we had that sort of 70% on average level of resistance. We know that you can still get control of cabbage stem flea beetle if you apply potentially in the evening where they're more active, because if you apply lambda at the rate we recommend onto the beetle, onto them, so you're actually hitting the beetle themselves, what's happening is that you're loading that individual beetle with a higher volume of product and you'll get control, because the metabolic resistance, which seems to be the predominant form of resistance cabbage stem flea beetle, can be overcome by the rate effect. So you will still get good control of adults at time of application. The level of control and residual effect will potentially go down over time with levels of resistance. It's hard to say, but you know, the caveat is that that was the level of control we're seeing this year, and that was the highest level of resistance we've seen across the country so far. So it's still having an effect, but we'll still try and develop the story and add those added elements, you know, like the cover crop. What else can we do to get good levels of control?
Brilliant, thank you, Max. Is there anything else that you're thinking of doing differently at the OSR site where you've been doing various different trials this year?
Yeah, so going forward, obviously we want to keep on innovating. So this year I think we've done very well with companion crop. I'm quite happy with that. The you know, AD digestate manure piece is because it's nuanced and will only fit if it fits the migration, and it only fits if you've got access to that and at the right timing. I think that's difficult to recommend. The straw, interesting, but I think will be hit and miss if it works or not. And the grazing is a bit difficult for all the yield losses we have seen attributed to it. So I think the next what we'll do is we're looking at more biological partners to help. So that will either be biostimulants to aid early establishment, either as seed treatments or foliar early in the season, We did try this year, and we tried a product which was looking at heat stress, because in previous years we've had the problem with drought and high heat levels in that August period. But like I was saying, this year we had basically, as soon as it was drilled, it was raining, and we didn't have those heat stroke events. So we might try that again. Then we're looking at what products can help that early vigor. Outside that, there might be certain things to help the penetration of lambda into the pest rather than into the plant, say. So that won't be an all, that'll be a different type of pest. And that might allow the benefit we see with direct application, because as I was saying, with direct application, you're technically increasing the concentration of lambda on the target pest, which overcomes the metabolite resistance because it gets a higher loading, that individual. So if you can do that by another means, that could be of interest. So those are the sort of things we're thinking about for this year. Establishment increases by vigor boosts, and then maybe adding other products in that could help the efficacy of lambda.
Do you think those kind of biostimulants will cross over into the BYDV sector as well, or will you be looking at different types in that area?
In the BYDV sector, similar types, but we've got a few other options in the BYDV sector, because we're also potentially exploring what we can do about the virus as well, whereas the beetle is very much about the pest itself. So we are expanding it and looking at other more innovative ways to control the pest, and it might be not so prophylactic.
Brilliant, thank you very much. Well, there's no further questions, or if no one wishes to raise their hand and ask a question directly, then we'll draw the webinar to a close. Thank you very much, Max. Very interesting and informative as always. And hopefully everyone has a good rest of their day. Thanks very much, everybody.